Thank you very much for kind introduction, Dr. Tan. It's my great pleasure to have opportunity to talk in this wonderful symposium. Today, I'm going to talk about the vaccine development of COVID-19. First of all, I'd like to start the brief introduction of SARS-CoV-2. Probably you may know that the SARS coronavirus is the largest RNA virus. It contained 30 kilobase single-strand positive sense RNA. The 2/3 of viral RNA directly translate into the 15 non-structural protein. And this protein cleaved by two proteases, NSP3 and NSP5, and into 60 protein. And NSP12 is an RNA-dependent RNA polymerase. And from NSP7 to NSP16, making a large replication complex. And this replication complex transcript the messenger, subgenomic messenger RNA coding for the structural protein, spike, envelope, matrix, and nucleocapsid, and other accessory proteins. So SARS-CoV-2 binding to the receptor, human ACE2, and then cleaved by TMPRSS2 and directly fused with the plasma membrane. This is one way to internalize. The other way to internalize is they bind to the receptor and go into the endosome, and then cleaved by patepsin, B or L, and then membrane fusion takes place between the endosome and viral protein. And then uncoating and viral RNA making a translation and oral replication takes place. So there is a two-way to internalize into the cell. So this is a particle, and then this is a spike protein. So spike protein bind to the receptors, as I mentioned, and the spike protein is a trimer. So this is the receptor binding domain, very important region for the interaction with receptors. So when you look at the monomer, the spike protein cleaved by furin and cleave into two parts, S1 and S2. And cellular protein, as I mentioned, TMPRSS2 or catepsin further cleave this region. And fusion peptide region, as shown in pink, are exposed. So this is a function of S protein. The particle bind to the receptors. Then, so S1 region released after binding to the receptor. TMPRSS2 or catepsin cleave this region and expose fusion peptide region. And this peptide fusion region bind to the host membrane, and S2 region here go to the conformational change, and finally making a 6-helix bundle, and viral RNA go into the host cells. This is an infection. So the vaccine for SARS-CoV-2, there is many vaccine candidates. The very classical one is attenuated live virus vaccine, and also inactivated vaccine, and viral-like particle vaccine, and purified spike protein vaccine, and also viral vectors vaccine. So, and the non-replicated viral vectors like adenovirus, and mRNA vaccine, and plasmid vaccine like this. These viral and nucleic acid vaccine making mRNA produce viral spike protein and induce immune response. So this is a candidate for vaccine, a little bit old, but the adenovirus vaccine and inactivated vaccine, mRNA vaccine, and S protein vaccine. For probably, as you know, this is AstraZeneca vaccine using chimpanzee adenovirus. And these inactivated vaccine mainly produced in China. So today I'm going to focus this mRNA vaccine produced by Moderna and BioNTech Pfizer. So probably you may know that these vaccines, Pfizer vaccine and Moderna vaccine, exhibit very high potency, more than 55% efficacy. So also, when we inject RNA into our body, these exogenous mRNA recognized by the toll-like receptor 7 or 8, and then induce very strong immune response. And also, so translation is suppressed. To overcome this immunogenicity of mRNA, Katarina Kariko, she is now in BioNTech, succeed in suppressing the immunogenicity of RNA by modifying the base to change the origin to pseudo origin. And this is the origin here, the pseudo origin like this, and also N1 methyl pseudo origin like this. So these modification of origin to methyl pseudo origin. So this is the origin. This is a methyl pseudo origin cannot recognize by the toll-like receptor 7 or 8 like this. So these RNA carrying the pseudo origin can escape from the innate immune response. So mRNA vaccine is just closing this S protein region, and then by using this DNA template, mRNA produced by using pseudo origin here, and then produce this is the S messenger RNA. They have the origin, pseudo origin in place of origin like this. And then this mRNA has, as I mentioned that, by using a pseudo origin, so we can suppress the immunogenicity of protein, mRNA, and also modify this 5-cap region to reduce immunogenicity. And also, they optimize codon sequence of S protein, and also enzymatic modified 5' region to inhibit enzymatic degradation, and also polymerase sequence improve the translation efficiency, and 5', 3' UTR region modification improved translation efficacy. And then pack into the lipid nanoparticle. This is the mRNA vaccine. So upon injection into cell, this mRNA vaccine, so that this mRNA directly translate into the protein. Some fraction go secreted into the out space of the cell, but some portion were degraded by the proteasome and then presented MHC class 1 peptide. And the secreted protein go into the cell again, and then presented class 2, MHC class 2 molecule. So by using this mRNA vaccine, we can induce antibody and also cell-mediated immunity as well. So this is the data of the phase 3 of BioNTech vaccine. As you can see by the single injection of our mRNA here, so the patient number is decreased, and then second shot, the completely protected. The efficacy is 95% compared to the placebo control. However, so probably you may know that there is an emergence of variants of SARS-CoV-2. This is the original Wuhan virus. And then quickly, D614G mutant, we called European type, appeared. And then UK variant, South Africa variant, Brazil variant, Indian variant called the Delta strain appeared like this. So this is the structure of the S protein monomer. And then it's the trimer. So this is the receptor binding domain. This is the N-terminal domain. This is gray is S2 region. And the receptor here. So to binding to the receptor S2, so receptor binding domain should be up like this. This is the down, it's the inactive home. To bind to the S2, the receptor binding domain should be up conformation. So the first mutation, D614G, is a European type, quickly appeared after the Wuhan outbreak. So just one mutation here. So this is a pseudotype bearing the spike protein of SARS-CoV-2 onto the vesicular stomatitis virus. So this is the original, compared to the original strain, this is a European type G614 mutant exhibited more potent infectivity. So why this enhancement takes place? This is the structure of the Wuhan strain here, and this is a G614G mutant. Compared to the original Wuhan strain, one mutation induces the number of the receptor up modification like this is increased. The original strain is only half of the receptor binding domain is open or up, but D614G, most of now 2 up or 3 up conformation is increased. That's why the infection efficiency increased. So this is D614G here. This is the European mutant. And then UK variant has a lot of additional mutation. And also this is a South African variant here. And also Brazilian variant having this mutation. And also when you look at N-terminal region here, UK variant, South African variant, Brazilian variant has a lot of the deletion or substitution here. So, but most important is the receptor binding region. This is a UK variant. As you can see, this N50Y mutation appeared. And this mutation also observed in the South Africa and Brazilian, but not Indian Delta variant disappeared. And also, South Africa has two additional mutation, and Brazilian is, this side is N2T here. And Indian variant, Delta variant, this N501 mutation disappeared. Instead, there is other mutation included. This is the summary of the variant of concern VOC, alpha, beta, gamma, delta. As you can see, this D614G is conserved, but 501 mutation is not in the delta strain, and the other mutation occurred in the various variants. So probably effectivity is increased the delta variant compared to the original Wuhan strain 2 times to 5 times. But we don't know the severity of the diseases. And also, even though these mutant current vaccine are useful to protect the infection. So, as you have mentioned, a lot of variants appeared. So, mutant virus appeared more and more in future, not only the Delta, but also Lambda, Mu, and so on. So, reverse genetics of SARS-CoV-2 is required for the analysis of mutant viruses. But coronavirus, as I mentioned, is the largest RNA virus. It's very difficult to construct the reverse genetic system. So we have to develop the simple reverse genetic. So far, we have a reverse genetic system for coronavirus by using bacterial artificial chromosome back. But it's very difficult because we have to amplify this huge back in the E. coli. So many mutations introduced during the amplification in E. coli. And the other method is in vitro ligation method. The cDNA ligate in vitro and then in vitro transcription and in vitro, and then transfect into the cells. So this system is very complicated and required masteries. So we use a very simple method called circular polymerase extension reaction. We call CPAR. We use this system is to construct an infectious clone of many flaviviruses, including hepatitis C virus. So we synthesize cDNA, just overlapping fragment, the cDNA fragment covering the whole sequence, and with promoter and ribozyme. And then after PCR reaction by using these fragments, we can directly transfect this PCR reaction into the cell. We can recover the infectious particle. So we use a lot of the flavivirus cDNA construction, but flavivirus is around 10 kilobases. It's 1/3 of the coronavirus. So no one tried to use this CPAR method to construct coronavirus. But so in my, Dr. Tori, she was in my postdoc, she challenged to apply this CPAR method to construct the coronavirus, SARS-CoV-2. She synthesized 9 fragments overlapping, covering the whole sequence of the SARS-CoV-2, and also promoter sequence of CMV and ribozyme sequence. And then did the PCR reaction. As you can see, a little bit long time, 25 minutes, like this, or 20 times. So finally, we succeeded to use this CPAR method to construct infectious clone of SARS-CoV-2. So upon transfection, you can see the very clear cytopathic effect. So by using these reverse genetic system, we can construct attenuated virus. and the reporter virus. And most importantly, we try to establish mouse-adapted SARS-CoV-2. So SARS-CoV-2 can infect human and monkey and hamster, but not into the mouse, but transgenic mouse expressing human ACE2, the virus can infect, but not in the wild type mouse. So we try to establish mouse adapted virus by using the CPAR method. So it's very old study is in case of SARS-1. So SARS-1 also utilized human ACE2. So transgenic mouse expressing human ACE2 can show the susceptibility to the SARS-CoV-1. But upon infection, this mouse showed encephalitis, not pneumonia. So upon infection from the nose, the most virus into the brain and very severe encephalitis. This is so it is not good model to the SARS-CoV-2. So as I mentioned, SARS-CoV-2 utilized human ACE2, but not mouse ACE2. So Ralph Baric group in US, so passage this SARS-CoV-2 in mouse, and then 2 amino acid substitution in the receptor binding domain, QP to YP. This virus can infect to the mouse. And also, they passage 10 times more. And then finally, this SARS-CoV-2 mouse adapt10, MA10 virus can replicate in the mouse. So we generated, published by the Ralph Baric's group, we construct MA10 virus. So this is a ACE2 receptor and receptor binding domain S protein. This is a human ACE2, and this is the receptor binding domain of S protein. But if you compare mouse and human, 3 amino acids are changed. So first, mouse adapted virus introduced 2 amino acid mutations. The further 10 times passage induced 1 more mutation. Altogether, 3 mutations are introduced in the receptor binding domain. Yes, so by 3 mutations, the virus can recognize mouse ACE2 as a receptor. So by using this mouse adapted virus, first we try to identify mouse basal dose 50. So this is a data of the body weight after 6 days post-infection. As you can see, 10 to the 1, 10 billion induce a little bit weight loss, but 100 virus, more than 100 virus induce very severe weight loss. And then this is a survival curve, as you can see. So more than 1,000, the virus can kill the mouse. So we calculate MLD 50 value. So this is a 10 to the 2.5 TCRD 50, around the 300 TCRD 50 particle can kill 50% of the mouse. Very, very severe pathogenic virus. So by using this mouse adapted virus, we try to assess the effectiveness of the vaccine we are now using. We try to produce viral-like particle by using insect baculovirus and also produce in the mammalian cell, human 293 cell. So this is VLP produced in the mammalian cell. This is the silver staining. So by using this viral-like particle, we immunize this viral-like particle carrying the D614G mutation and with adjuvant, alum oil or saponin, and inject 2 times like this. And then 4 weeks after the second dose, we inject 10 to the 4th TCID50 mouse adapted virus, and then examine the body weight loss, antibody titers, and viral titers. This is immune response determined by the ELISA against S protein. As you can see, first dose, we can see the induction, the immunized mouse with adjuvant, oil adjuvant or saponin, but not without adjuvant. After second dose, we can see the antibody response in alum adjuvant, oil, and the saponin, but also only the VLP without the adjuvant, like this. And then this is the body weight loss after challenge of the mouse adapted virus. As you can see, the mice immunized with various adjuvant, so body weight loss recovered, but not in the VLP alone. And this is the viral titers in the lung of the vaccinated mice challenged with M810 mouse adapted virus. As you can see, 3 days and 6 days post-infection. As you can see, by the immunization of the VLP with oil saponin adjuvant, you can see the very good protection. These immunizations suppress the viral titer in the lung compared to the alum adjuvant or without adjuvant. Then finally, we examine the survival rate of the vaccinated mice after challenge with mouse adapted viruses. As you can see, so all mice immunized with VLP with alum, oil, saponin completely protect the lethal challenge of the mouse adapted SARS-CoV-2, and also VLP alone shows some protection. So these data indicate that the VLP we produce by using European type virus can block the challenge of mouse adapted virus. Okay, so these data indicate that this VLP is a very potent candidate for the subunit vaccine of SARS-CoV-2. So this is data. This work was mainly done by Chikako and Itsuki, Chiho, Takasuke in our laboratory. Thank you very much for your attention. Sorry to delay